Welcome dear students. In this video we are going to learn about malate-aspartate shuttle. This shuttle is related with cellular energetics. But before I go to the topic, I want to ask a very general question. Why this shuttle is required for a cell? And the answer actually relies in the glycolysis. As we all know that glycolysis occurs in the cytoplasm and it produces 2 ATP and 2 NADH molecules. Now these NADH molecules is the reason for this malate-aspartate shuttle. It's actually a bypass. So what is happening here? NADH is actually high energy electron carrier. One NADH molecule can carry 2 high energy electron, and it actually eventually take part in the electron transport chain and helps in the generation of ATP indirectly. This is the simple role of NADH. Now the problem is here in the cytoplasm, the NAD plus is actually reduced to NADH. Now to continue the glycolysis process, a continuous supply of NAD plus should be there in the cytoplasm. But cytoplasm has a limited amount of NAD plus. So it should regenerate the NAD plus from the NADH. And cytoplasm relies on this mechanism on mitochondria. But there is a problem that the mitochondrial inner membrane is actually impermeable for both these two products. So let me draw the inner mitochondrial membrane, because here this shuttle actually happens between cytoplasm and the matrix, and this inner mitochondrial membrane is the barrier for these two molecules to exchange between the cytoplasm and the matrix. Now this is the matrix of the mitochondria and this is the cytoplasm. Let's suppose. So basically this shuttle actually a bypass mechanism by which, as these two molecules cannot enter inside the mitochondrial matrix and cannot directly take part in the electron transport chain, it exchange its electron through some molecules present in the cytoplasm. But as it is a shuttle or is a cyclical process, so there is no starting point and end point. But for our convenience to understand the mechanism, we will start from a molecule called oxaloacetic acid. Let's suppose. So this oxaloacetic acid is actually converted, this is the first reaction that takes place, is converted into malate. And the enzyme that is responsible for is called malate dehydrogenase. In my TCA cycle lecture, I have already discussed about how malate and oxaloacetate are reversibly converted, can be converted, and the enzyme, wherever you find the name dehydrogenase, that means it can exchange hydrogen. So what happens here? One NADH molecule actually oxidized to NAD plus. So you can see here the NADH that is generated from the glycolysis are actually release its high energy electron and the hydrogen to the oxaloacetic acid, and then oxaloacetic acid is converted into malate. This is a simple fact that occurs. Now in the mitochondrial matrix, I have mentioned during TCA cycle that the malate can be converted into oxaloacetic acid. But in the mitochondrial matrix, what happens? NAD plus actually converted into NADH. Now you can see the opposite is actually happening, and this is actually the reason, because oxaloacetic acid now becoming the malate, so the NADH actually oxidized to NAD plus and it actually release the high energy electron and the hydrogen to the malate. And the same enzyme here also, the malate dehydrogenase perform the functions, here also malate dehydrogenase perform its function. But this cytoplasmic malate dehydrogenase is a version of the matrix malate dehydrogenase. Now in the inner mitochondrial membrane there is a antiporter. There is an antiporter. This antiporter is called malate alpha ketoglutarate antiporter. As you can see, this shuttle is called malate-aspartate. So this malate is the first product that actually enter inside the mitochondrial matrix. So malate enter. Now, as I have mentioned, this is the antiporter. So when a malate enter, one alpha ketoglutarate should come out from this matrix. So one alpha ketoglutarate actually come out from the matrix to the cytoplasm. This is a antiporter. Now the malate, when enter into the matrix, it actually gets converted. It gets converted into oxaloacetic acid by the same enzyme malate dehydrogenase that is present in the matrix. Now the opposite thing occurs. What happens here? NAD plus that is present in the mitochondrial matrix are actually reduced, actually carry the high energy electron to become NADH. So you can see here, this is actually a bypass mechanism by which, as the NADH cannot enter inside the inner mitochondrial membrane or the matrix, so it actually bypass through the malate, and this malate becomes oxaloacetic acid. Then it actually transfer the high energy electron to the NAD plus that is present in the matrix. Now this oxaloacetic acid, it get converted into another product, and it's a reversible process. The product is called aspartate. That is why it's called malate-aspartate. Aspartate. So the enzyme name is called transaminase. Okay, now I will draw the chemical formula so that you can understand these three molecules, aspartate, oxaloacetic acid and malate, very easily, and you can understand the mechanism. Because in the transamination process here, what happens, I will show you. At first I will draw the chemical formula. The aspartate chemical formula is C double minus CH NH3 plus CH2 COOH. This is the chemical formula of aspartic acid or aspartate. Aspartate. Now, as I have mentioned, that this oxaloacetic acid can also actually generate from aspartic acid. So let me draw the antiporter, then you can understand. This aspartate actually shuttle. So the malate and aspartate actually shuttle between cytoplasm and matrix. The aspartate come out from the antiporter. This is the antiporter. This antiporter name is, here is the malate alpha ketoglutarate antiporter. The antiporter is called aspartate and glutamate antiporter. So there are two antiporters that take place in this shuttle. So one aspartate actually come out into the cytoplasm. Now the aspartate is actually converted into oxaloacetic acid, so it's the cyclical mechanism. Okay, and the same enzyme, the transaminase, actually take part in these reactions. But one thing occurs, you have to understand this mechanism and you can understand this chemical formula. The aspartate contain a amino group, and this transaminase actually release the amino group to make oxaloacetic acid. So the structure of the oxaloacetic acid should be very similar to the aspartate, like it is C double minus C double bond O CH2 C double minus. You can see here, only the difference is here that it has no amino acid and it has a keto group. So this is actually oxaloacetic acid. Now I have mentioned that oxaloacetic acid can be converted into malate by exchanging hydrogen. So the structure of oxaloacetic acid and malate is very similar. It is C double minus CHOH CH2 C double minus. You can see here is a hydrogen, and here is hydrogen actually accepted from this NADH, and it actually carry the high energy electron that is actually donated through this process. So it is actually malate. So you can see here the structure of these three molecules is so similar. Now this is actually the shuttle that actually takes place. Now the story comes here for the glutamate and alpha ketoglutarate, because I have mentioned this is the antiporter if one malate to come inside the matrix, one alpha ketoglutarate should come out, and if one aspartate come out from the matrix, so one glutamate should come in, glutamate. And here the very important process occur, and that is this glutamate actually have a amino group that is actually exchange with this oxaloacetic acid, and this oxaloacetic acid actually become the aspartate. You have to understand this. You can see here the oxaloacetic acid don't have the amino group. So where from this amino group comes, where it is converted into aspartate, the transaminase enzyme actually take the amino group, okay, take the amino group to the oxaloacetic acid to make it aspartate. Now when glutamate actually release the amino group, it actually become the alpha ketoglutarate. So you have to understand this. So when this glutamate release the NH2 group, it becomes the alpha ketoglutarate. Now the glutamate actually enter in the cytoplasm, okay, in the cytoplasm, and this glutamate actually take this amino group, because aspartate to make oxaloacetic acid it should release the amino group. Now the glutamate take this amino group to make glutamate. Sorry, the alpha ketoglutarate actually take the amino group to make glutamate. Very easy, you can remember this. So this is the source where from this glutamate is coming. So you can see it is actually a cyclical process. Now let me draw the structure of glutamate and the alpha ketoglutarate so that you can understand easily. Okay, here the structure of glutamate is like this: CW minus CH NH3 plus CH2 CW minus. Okay, this is the structure of glutamate or glutamic acid. So you can see here is a one carbon CH2 group is extra, and there is a aspartate and a glutamate. The structure is very similar. Now when the glutamate becomes alpha ketoglutarate, what happens? The structure is very similar. It becomes like this. This is the structure, molecular structure of alpha ketoglutarate. So when a glutamate amino acid is converted into alpha ketoglutarate, it just release the amino group. So when glutamate entered inside the matrix, it released the amino group and it becomes the alpha ketoglutarate. The alpha ketoglutarate come out from the matrix because it exchanged with this antipoda and actually take the amino group from the aspartate, because the aspartate actually becoming the oxaloacetic acid, and then it actually becomes the glutamate. That glutamate actually enter into the matrix. So this is a subtle mechanism here. Malate and aspartate actually subtle, and by the way, the NADH that actually produced from this glycolysis, that is carrying the high energy electron, are actually bypassed through this molecule, and ultimately generate one NADH molecule inside the matrix that directly take part in the electron transport chain and helps in the generation of ATP. Now this mechanism, Malate-Aspartate subtle, actually occurs in the cardiac myocytes, cardiac muscles, and the liver. But in our skeletal muscle, there is another type of subtle mechanism is there. We call glycerol 3-phosphate. In my next lecture video, I will make a separate class on glycerol 3-phosphate subtle. So hope this video will help you. Thank you for watching, and always keep learning.